fun of my headphones. These are nice headphones. Okay, how do I look? Space Cadet? Okay. All right, so Unit 5, Traits Are Us, Review for Unit Test. You can find this review and this video at srhsbio.wikispaces.com. First section is about protein synthesis. So how can we use the code in DNA to make proteins and influence the cell? First set of questions is to label the following. So first we have DNA. You find DNA in the nucleus. The DNA is used to make an mRNA code. That's transcription. It is unwound by an enzyme. The mRNA is made. After it has been made, the mRNA is going to exit the nucleus and it will enter the cytoplasm. Inside the cytoplasm, the mRNA will meet up with a ribosome, and ribosomes are also made of rRNA. tRNA is the molecule that brings the amino acid to the ribosome mRNA complex. So we see these three prongs that we have on the tRNA. Those three prongs match up to the mRNA down here. We then attach amino acids using the ribosome. And once we link a bunch of amino acids together, we make a protein. This is all done inside of the cell, so around the edge of the cell, that is the cell membrane. Now let's look at transcription. So we're going to zoom in on the part of the cell where the nucleus is, and this is where transcription occurs. So DNA is the original blueprint. It is unzipped by an enzyme so that I can make a working copy. That's the mRNA. So we can think of the DNA as being our original blueprint and the nucleus as being City Hall. We store the DNA where it is safe in City Hall, in the nucleus. If you were doing work on your house, you would need to go into City Hall and make a copy of your architecture plans. So we keep the DNA safe in the nucleus by making a copy of it and taking that copy with us instead. DNA, if we have ACC, AGC, that's going to become an mRNA, UGG, UCG. So big thing to remember is that in RNA, there is no thymine, there is no T. Instead of T, we have uracil. Translation occurs in the cytoplasm of the cell. So first, the mRNA needs to exit the nucleus, and it's going to attach to a ribosome. The ribosome is going to read the mRNA in sets of 3. So each 3 mRNA bases represents a codon. Each tRNA matches that codon. So each tRNA molecule has an anticodon that matches the codon in the DNA. So this is how we know that we're bringing the correct ribosome. Only the one with the right anticodon is going to fit the codon of the mRNA. And the ribosome knows if it doesn't fit, that's not the right protein. So the amino acids are linked together and they make a protein chain. So if the mRNA was UGG, UCG, the amino acids would be tryptophan and serine. Next is about the role of mRNA, tRNA, and rRNA. And if you understand what these things do, then you understand protein synthesis. So mRNA is what we call the messenger mRNA. And the reason we call it is because it's carrying DNA's message. It's carrying that code from the nucleus out to the cytoplasm so protein synthesis can occur. tRNA is the transfer RNA. Its job is to bring the correct amino acid to the ribosome. We know which one is correct because it has an anticodon that matches the codon of mRNA, and it brings the amino acid. The rRNA, so the R is for both reads and ribosomal. So the ribosome is made of rRNA and a couple other proteins complexed together. So that complex of rRNA and proteins makes up the ribosome. Next question, what is the relationship between genes, proteins, and traits? So here we see that we have a DNA code, and that DNA code codes for proteins, and we make those proteins with protein synthesis, the process that we just outlined. Each of these proteins affects cell function, and it leads to the expression of traits. So first we have DNA that codes for structural proteins. Structural proteins lead to your physical traits, so your hair color, your eye color, your skin color, those type of physical traits. Enzymatic proteins. These are proteins that affect your metabolic traits. So for example, lactose intolerance would be an example of a metabolic trait. You're unable to make that enzymatic protein, so it affects your metabolic processes of digesting milk. And hormonal proteins. These are proteins that control growth and development of an organism. So these different types of proteins are all coming from DNA's code, and they lead to different traits. So DNA codes for proteins, proteins lead to traits. That's the relationship between those three pieces. Next question. Human body cells all have the same DNA. They have the same genetic material. Consider a human heart cell and a human liver cell. How can these cells perform such different functions but still all have the same DNA? So, different genes, different segments of DNA are going to code for different proteins, and there's different ones turned on in each cell type. So every single cell has all of the same DNA, but not all of those genes are active in every single cell. A heart cell is going to have different genes active than a liver cell. Because they have different types active, it's going to lead to different proteins, which means different types of cells. So the difference between a heart cell and a liver cell, it's just that there's different proteins being made because different genes are active. This leads to all the different cell types. So up here we've got connective tissue and nervous tissue and cardiac muscle. These all look different. They're expressing different traits. They're expressing different proteins, different genes. Next question is about the relationship between DNA, genes, and chromosomes. So here we see DNA, and that DNA is tightly wound into a chromosome. Now, just before a cell gets ready to divide, those chromosomes condense. It's kind of like they're packing up, getting ready to move during cell division. So before cell division, the DNA coils up into tightly bound chromosomes. The genes are segments of DNA that code for a specific protein, and each segment of the chromosome contains the DNA for a gene. So, chromosomes, tightly bound DNA. Genes are made of DNA. Number 5, what do the bands represent on the chromosome? So, each segment of DNA corresponds to a gene, and we can identify and label those genes with banding. Each band corresponds to a specific gene. So do chromosomes actually have bands on them? No. Scientists can color code and label a chromosome that they are studying so they see where the genes are on the chromosome. Next section is about mitosis and asexual reproduction. So how do cells duplicate? So first question asks us to consider and explain the process below. So this is the process of mitosis. There are 4 phases in mitosis, thus there are 4 pictures of the cell. First is prophase, and we call it prophase because the cell is preparing by duplicating its DNA. So in picture 1, we see that the DNA is already duplicated. Next, metaphase. In metaphase, we meet in the middle. So those duplicated chromosomes have lined up in the middle of the cell. Next, anaphase. They're going to start to move away from each other. So the spindle fibers, these guys here, are connected to each half of the chromosome. And then whenever we get to anaphase, they pull away, pull the two halves of the chromosome apart. And the last phase, which is in picture 4, that is telophase. So in telophase, that is where they're going to split into two. They're going to evenly divide the organelles. So if there's mitochondria or chloroplasts, ribosomes, all of those things get evenly divided, and the cytoplasm also gets evenly divided. The next question asks us to comment on the genetic similarity between the parent cell, which is cell 1, and the daughter cell, cell 4. So in cell 1, we see the DNA is already duplicated. So it might look like cell 1 and cell 4 have different chromosome numbers. They don't. It's just that in cell 1, we've already duplicated. So if we were to look at cell 1 before duplication, it would look like this. So here we see that we have 3 chromosomes. The parent and daughter cells are identical. They have 3 chromosomes, same chromosome number in each. The reason for this is because mitosis is simply the process that we use to duplicate cells. They're going to be identical copies, clones. So if you are a single-celled organism, this is a way that you can reproduce yourself. If there's one bacteria and it splits apart into 2, that's reproduction. So for single-celled organisms, mitosis is just a type of asexual reproduction. The next question asks us about the relationship between cancer and mitosis. So we know that mitosis is cell division. Now, in order for cell division to occur, this is a very carefully regulated process by the cell. And there are certain checkpoints that a cell must pass in order to duplicate. If it doesn't meet those checkpoints, it doesn't duplicate. It won't divide. In a cancerous cell, that machinery, those checkpoints have gone haywire. It's bypassed all of the checkpoints. So it's just going to divide and divide and divide and divide without any regard to the normal cell cycle. So cancer is just unregulated, haywire cell division. And cell division is just a way of saying mitosis. So the relationship between cancer and mitosis, cancer is mitosis. It's just mitosis that's gone haywire. Too much cell division. Next question asks us about types of asexual reproduction. So first kind we have is budding. So this is what we see in the hydra, which is a little single-celled pond creature. The hydra has a little tiny clone that grows off the parent, and once it's fully matured, it falls off. So imagine you've got a little mini version of yourself growing off of your side, and then one day, little mini you falls off and runs away. Vegetative. So it's got the word vegetable in it. So this is something that plants do. Potatoes, strawberries, vegetables. This is where shoots are going to grow off of the parent, and then they're going to develop into independent clone plants. Last kind is binary fission. I remember this because they both start with B, binary fission, and bacteria. So this is how bacteria reproduce. It just duplicates its DNA and splits into 2 cells, just mitosis. All of these types of asexual reproduction, they all result in clones of the parent. So there is no genetic diversity here. They are all going to be the same as their parent cell. Next section is meiosis and sexual reproduction. So how does meiosis create unique gametes in order to allow for sexual reproduction to occur? A human somatic cell has 46 chromosomes. So there's some vocabulary in this question that you have to be familiar with. How many chromosomes are in a human gamete? So a somatic cell is the same thing as a body cell. Those have 46. Gametes always have half as many chromosomes as the body cell. So if this body cell has 46, the gamete or the sex cell must have 23. So you have to be familiar with those words. You have to know that somatic cell and body cell, two words for the same thing. Gamete and sex cell, different words for the same thing. Next, what is the ratio of chromosomes in a somatic cell to a gamete in a human? So a somatic cell, 46. Gamete, 23, which reduces to a ratio of 2 to 1. And in fact, that is the ratio for all organisms, because we need 2 gametes to come together to make 1 zygote. Crossing over, how does it contribute to sex cells that are genetically different from their parent cell? So here we see crossing over occurring. 2 sister chromatids during meiosis 1, they're going to line up next to each other. So these guys will line up next to each other. And then they're going to cross over their DNA. And once they do this, we end up with these chromosomes that look like little patchwork quilts of the other chromosomes that they crossed over with. This leads to more genetic variation while making gametes. So this is just one more way that we get gametes that are different from the parent. So just to illustrate that, here is a cell that is an interphase, and this cell is going to be used to make gametes. So here are the gametes that are produced with crossing over. Here are the gametes that are produced without crossing over. So we can already see that there's a difference between the two. Crossing over is going to lead to more unique combinations, more possibilities. And this picture is just showing us where it's crossed over at one point. So imagine if they crossed over at many points. There'd be many, many more combinations possible. Benefits of sexual reproduction. The biggest one is genetic variation. It allows the parent and offspring to be genetically different from each other. This helps to ensure the survival of the species. Organisms do not all have the same weakness, so it's harder to wipe them out. So if we were all genetically the same and we had weakness to some disease, if that disease came around, it would wipe out our whole species. But because we are genetically different, our species has a better chance of survival. Number 12 is to label the angiosperm. So the anther, that's where the pollen is made. So if you look at a flower, those are the yellow parts. If you touch them, the pollen rubs off on you. The anther is held up above the petals by the filament. The reason for this, you want that pollen to be easily available to pollinators, bees, bugs, wind, because we want that pollen to be spread around. So it's held above the flower, so if a bee comes by or a hummingbird or a nice breeze, that will spread around the pollen. At the end of the sticky stamen, so this is the stamen and this is the style. At the end of that, we find the ovule. And the sepal, those are like extra petals that protect the ovule and hold the petals on the flower. The petals are bright and colorful to attract those pollinators as well. So you need to know where meiosis is taking place in this angiosperm. So first, the anther and the ovule. So the anther makes pollen. That's kind of like plant sperm. The ovule makes the ova, which is a plant egg. When that egg is fertilized by the pollen, you're going to form a seed. And many different plants protect their seeds in fruit. This is so that animals come and eat the fruit and spread those seeds through their droppings. The next section is about genetics. So how do genes result in traits? So an example of a phenotype. So phenotype is a physical characteristic. They both start with pH. So tall, short, those are physical characteristics. Those are phenotypes. A genotype. So genotype is based on genes. So big Q, that's a genotype. ATC, CGA, ACTC, this is a DNA sequence. This is also a genotype. Hetero means different. So heterozygous would be big T, little t. Homo means the same. So that means the same. So big T, or big T, little t. Excuse me, little t, little t. Next question. A group of scientists were studying the gene for eye color in fruit flies. They began by mating pure breeding white-eyed females. So they mated a pure breeding white-eyed female with a pure breeding red-eyed male. So pure breeding means that it is homozygous. Then they take those offspring, so those result in the F1 generation, and they're going to cross those two in order to make the F2 generation. So first question, use the data to explain why eye color must be sex-linked, a trait in fruit flies. So males inherit their X chromosome from their mothers. What that means is that we see that all of the males end up being white-eyed because they all inherited that gene from their mother. All the males in the F1 generation, they inherited that trait from their mother. Thus, eye color is sex-linked because it is inherited differently for males than it is for females. If this were non-sex linked, that F1 generation would all show the dominant phenotype. So whenever Mendel did this with his pea plants, he mated a tall plant, big T, with a short plant, little t, and the first generation was all big T, little t. They were all tall. So we don't see that. We still see, if it were non-sex linked, that F1 generation would be all red-eyed. The fact that it is inherited differently for males and females tells us that it must be sex-linked. So we know that red is dominant. We know that by looking at the females. If we look at the females, all of the white-eyed trait, that disappears because all of the females are heterozygous. So the females are heterozygous and they're red, which means red must be dominant. So for the parent generation, we took a pure breeding red-eyed male and we took a pure breeding white-eyed female and we crossed them. So pure breeding red-eyed male. So his sex chromosomes are X and Y and his eyes are red, so that's big R on the X chromosome. The female was pure breeding white-eyed, so her sex chromosomes are X and X, and she's pure breeding white, which means little r, little r. So whenever we mate these flies together, here is a female that is heterozygous for red eyes. Here is another female that is heterozygous for red eyes. So this is what we saw in our F1 generation. In the F1 generation, we saw that 100% of the females were red-eyed. They were all heterozygous. Now let's look at the males. So remember, in order to be a male, you got the Y chromosome from your dad, and the X chromosome comes from your mom. So since the females are homozygous recessive, the males are going to inherit a recessive gene. So we end up with X from mom, little r, Y, there's a male with white eyes, XR from mom, Y, there's another male with white eyes. So this is what we saw. 100% males were white-eyed. So this in here, these guys in this box, this represents my F1 generation. Okay, now let's make the F2. So I need to take these flies, a brother-sister fly, and mate them together. It's okay, they're flies. So, the white-eyed male. The white-eyed male has to be X little r y. And the female is going to be heterozygous. So what we did is we took this fly, I took this male, there he is, and I took this female, there she is, and now we're going to breed them together. So here is a heterozygous red-eyed female, a homozygous recessive white-eyed female, a red-eyed male, and a white-eyed male. So if the gene is sex-linked, there's only going to be one gene for males. So they're always homozygous because they only have one. So these flies that we produced in this crossing, these flies represent our F2 generation. Next question, the genotype and phenotypes of the females. So females are both going to be XX. The red-eyed female, F2 females. In the last slide, we saw that the female that is red-eyed has to be heterozygous. So for this one, the genotype is big R, little r. For the white-eyed females, you can only get those with little r, little r. So again, the way we got that, we took our white-eyed male, our heterozygous female, there's my heterozygous red-eyed female, there's my white-eyed female. The next question has a pedigree for flat-footedness. So it says that the allele for normal foot arches is dominant to having flat feet. In the pedigree below, label each individual with their genotype. So we know that big A is going to be normal, and little a is going to be flat feet. So let's start with the easy ones. The ones that are shaded in have flat feet. I know that they must be little a, little a. All right, so now let's start up with generation 1. If you look at generation 1, they do not have flat feet, which means they could be big A, or big A, little a. But if you look at their children, they have a child who does have flat feet. So it's impossible for them to both be big A, big A. The only way that those parents can produce an offspring that is flat-footed is if they are heterozygous. So these parents must be heterozygous. Now, if we look at generation 2, first let's look at individual 4. So individual 4, if they were big A, they could be big A, or big A, little a. If they were big A, they couldn't have children who are little a, little a. because one of those big A's would always be inherited by their child. So this man, individual 4, can't be big A because he has children who are flat-footed. So he must be big A little a. That's the only possibility that will allow him to have flat-footed children. All right, so then if we were to cross big A with little a, we would end up with either big A or little a. So offspring here, generation 3, offspring 4, also must be big A little a. Offspring 6, generation 3, must also be big A little a. So if we look at individual 7 in generation 3, this individual could be big A or big A little a. They can't be big A because then all of their children would have to be normal arches. Because we see that we have little a, it must be heterozygous. Otherwise, there's no way that this man and this woman could have offspring that are little a. All right, now let's look at this side over here at generation 2, individual 1. So this individual, they have 2 options. They could be big A, or they could be big A little a. Their offspring, we don't know whether or not this individual 1, we don't know what that person is because they've only had 2 children. If they had 15 children and none of them had flat feet, then it would be big A. If they have a 3rd child and that 3rd child has flat feet, then they must be big A little a. Now, as far as the offspring, we know that mom is little a. So all of these offspring so far, because they get one from their mom and the other one from their dad, they have to be big A little a. They can't be little a because they're not shaded in. And they can't be big A because they get one of their A's from their mom. So these guys must also be heterozygous. So this is like a puzzle. You got to start with the information that's obvious, which ones were little a, and then work your way backwards. If you were asked a question only about generation 2, individual 4, if you were only asked about that one, that's the only one that you should figure out. You don't have to figure out the whole pedigree unless you're asked questions about every individual in the pedigree. Next question. Use a Punnett Square to explain why you are unsure about the genotype of individual 2-1. So we just went through that. We know that the offspring must be big A little a. We know that mom is little a, but we're not sure which one this is going to be. So we don't know. They could be big A or big A little a because their parents are heterozygous. If 100% of individual 2-1's children have normal arches, then they must be big A. So right now they only have 2 children. So if they have 10 more children and they're all normal footed, then the dad is probably big A little a. If any of the children have flat arches, then this man must be big A little a. Otherwise, it's impossible for his children to have flat feet. They just have to have more children for us to be sure. So if we were to assume that the male was big A, mom is little a, so we would see 100% of the children with normal arches. They would all be heterozygous, but the phenotype that we would see would be normal feet. If the man was big A little a, we already know that mom is little a. So if that were the case, we would see on average 50% of their children with normal arches and 50% with flat feet. So maybe child 3 has flat feet. Maybe child 3 has normal feet and we don't learn anything. Next, individual 3-7. Why must they be heterozygous? So we know that individual 3-6, we know that they must be big A, little a. And we know that because they're parents. Their mom is little a. Dad has to be heterozygous. So we know individual 6 must be big A, little a. Individual 7 cannot be big A. Otherwise, it wouldn't be possible for this individual to have children who are little a. So they must also be big A. So if we do a cross, there is homozygous normal feet, heterozygous normal feet, heterozygous normal feet, and flat feet. So the only way for this marriage between individual 6 and 7, the only way for them to get children who are little a is if they are both heterozygous. Okay. Anything you want me to go back to?